Right now, somewhere beneath you, there is a world that no human being has ever seen. Not explored in limited detail, not partially mapped, never seen at all. The ocean covers 71% of this planet. It is the dominant feature of Earth, visible from space as the blue that defines us. And yet, we have explored less than 5% of it. The remaining 95% is as unknown to us as the surface of a distant planet. More unknown, actually. We have better maps of Mars than we do of our own ocean floor. Think about what that means. Mars is 140 million miles away. We have sent robots there, photographed its surface in extraordinary detail, analyzed its soil, mapped its craters and canyons with precision that would embarrass our ocean surveys. The ocean is right here, surrounding every continent, accessible from every coastline on Earth. Its deepest point is closer to us than many major cities are to each other, and we know almost nothing about what lies in its depths. This is not an exaggeration. This is the official position of the National Oceanic and Atmospheric Administration of NASA, of every major scientific institution that has studied the question honestly. The deep ocean is, by any reasonable measure, the largest unexplored territory on this planet, and it has been waiting in darkness at pressures that would crush a submarine for billions of years. We talk about exploring space as the final frontier. We celebrate the images returned by the James Webb Space Telescope, images of galaxies so distant that the light reaching our instruments left those galaxies before the Earth existed. We marvel at the photographs taken on the surface of Mars, images of a world 140 million miles away, captured by robots we built and landed with extraordinary precision. We point our telescopes at the moons of Jupiter and find evidence of subsurface oceans. And then we look at the floor of our own ocean and see almost nothing, because we have never looked. If you enjoy drifting into the deep ocean and discovering what lies beneath, please consider liking and subscribing. It truly helps more than you know. Are you ready? Let's begin. Ask anyone why we haven't explored the ocean, and they will give you an answer with confidence. It's too deep, too cold, too dark, too expensive. The pressure would destroy any vessel we could build. The distances are too vast. The technology doesn't exist. Every one of these answers contains some truth. None of them explains why 95% of the ocean remains unseen. The real explanation is stranger. It is more uncomfortable. It comes down to an assumption that shaped centuries of scientific thinking, an assumption so deeply embedded in how we thought about the ocean that it became invisible. And that assumption was this: the deep ocean is empty. Not metaphorically empty, not sparsely populated, empty. A cold, dark, featureless abyss where nothing significant could possibly live, nothing worth studying could possibly exist, and nothing important was happening. That assumption was wrong in the most profound way an assumption can be wrong. It was wrong not at the margins, but at the center. The deep ocean is not empty. It is not featureless. It is not a dead zone. It is the largest living space on this planet, occupied by organisms we cannot yet count, running on energy systems we are still learning to understand, structured by geological processes that shape the chemistry of every ocean and ultimately the chemistry of our atmosphere. We ignored it for centuries because we assumed there was nothing there to find. We were catastrophically, embarrassingly, wonderfully wrong. To understand why this matters, you need to understand the scale of what we're talking about. Not just the depth, not just the pressure, but the sheer volumetric immensity of the ocean as a living space. The ocean covers approximately 361 million square kilometers of the Earth's surface. Its average depth is approximately 3,700 meters. Multiply those numbers together and you get the total volume of the ocean. Roughly 1.335 billion cubic kilometers of water. Written another way, that is 1,335 million cubic kilometers. A cube of water with sides 1,100 kilometers long. More than enough to drown every mountain on Earth and still have water left over. The ocean contains 97% of all the water on Earth. It contains 97% of all the water in Earth's biosphere. Every lake, every river, every glacier, every aquifer, every drop of moisture in the atmosphere, everything that makes Earth the water world we see from space represents only 3% of the total. The rest is in the ocean. Now consider depth. The ocean is not a shallow pond that happens to be very wide. Its average depth of 3,700 meters means that if you placed a typical skyscraper at the bottom of an average location in the ocean, it would be completely hidden under more than 3 kilometers of water. The ocean has places where it is 5 times deeper than that. Places where the seafloor has never been touched by sunlight. Not once in the entire 4 billion years the ocean has existed. The vast majority of this volume lies in perpetual darkness. Sunlight penetrates seawater with steadily declining efficiency. The uppermost 200 meters, called the euphotic zone, receives enough sunlight for photosynthesis. This is the thin, sunlit skin of the ocean, the part we see in photographs of tropical reefs and surface waters, the part that contains most of the ocean life that has been studied in any detail. Below 200 meters, light begins to fail. By 1,000 meters, all visible light has been absorbed. Below that depth, which oceanographers call the midnight zone, there's no sunlight at all. None. Not a photon, not a hint. Perfect, absolute, permanent darkness. The midnight zone and the abyssal zone below it account for the vast majority of the ocean's total volume. The sunlit upper ocean, the part we know something about, the part that has been photographed and sampled and observed in detail, is a thin surface layer compared to the dark water below. This is what the unexplored ocean actually is. Not a dramatic trench or an inaccessible chasm. The ordinary, deep ocean, the mile after mile of dark water that fills every ocean basin between the continental shelves and the deepest trenches. The abyssal plains, the mid-ocean ridges, the seamounts, the cold seeps, the vast stretches of sediment-covered ocean floor that extend across areas larger than any continent on Earth. This is what 95% means. Not 95% of some dramatic, extreme environment. 95% of the entire ocean, most of it perfectly ordinary by deep-sea standards, simply never seen. The continental shelves, the shallow areas bordering most coastlines, represent a small fraction of the total ocean floor. They are well explored, relatively speaking. Fishing industries have dragged nets across much of them. Research vessels have sampled their sediments. Offshore oil and gas operations have mapped their geology in extraordinary detail for commercial reasons. The continental shelves are not the unknown. Beyond the shelves, the seafloor drops away. At the shelf edge, typically at depths of 100 to 200 meters, the bottom falls steeply away in what oceanographers call the continental slope. In some places, this drop is gradual, taking hundreds of kilometers to descend several thousand meters. In other places, it is almost vertical, a cliff of rock and sediment plunging into darkness within a few kilometers of horizontal distance. At the base of the continental slopes lies the abyssal plain, flat, covered in fine sediment that has been falling from the surface for millions of years, extending in some ocean basins for thousands of kilometers without significant relief, deeper than any structure on land, darker than any cave, colder than almost any environment humans encounter, and almost entirely unexplored. The abyssal plain is the floor of the deep ocean. It is where the bulk of the unexplored 95% lies, not in dramatic trenches, though those are unexplored too. In the quiet, flat, sediment-covered floor of the ordinary deep ocean, in the places that seem from the surface too boring and too inaccessible to be worth studying. They are not boring. They never were. This is what the assumption of emptiness cost us. Not just knowledge about exotic extreme environments, knowledge about the floor of our own ocean. The most common seafloor environment on Earth, covering more surface area than all the continents combined, remains largely a mystery because we assumed there was nothing important happening there. The mid-ocean ridge system provides the most dramatic illustration of how wrong that assumption was. Stretching approximately 65,000 kilometers around the planet, the mid-ocean ridge is the longest mountain range on Earth. It winds through every ocean basin, connecting at various points to form a single continuous chain of underwater mountains that circles the globe like the seam of a baseball. It runs down the center of the Atlantic Ocean. It curves through the Indian Ocean. It crosses the Pacific. In total, it covers more linear distance than any surface mountain range by a factor of many times. This is not a chain of small hills. The mid-ocean ridge rises several kilometers above the surrounding ocean floor in many places. Its peaks, the points of highest elevation along the ridge crest, sit at depths of roughly 2,000 to 2,500 meters in most of the Atlantic. Deep enough to be in complete darkness, but shallow enough that the pressure is not extreme by abyssal standards. Some ridge sections are so high that their peaks actually break the surface, forming volcanic islands. Iceland is a piece of the mid-Atlantic ridge that has built itself above sea level through accumulated volcanic activity. The mid-ocean ridge is where the Earth's tectonic plates are created. At the ridge crest, the plates on either side are being pulled apart by convection currents in the Earth's mantle. As they separate, molten rock wells up from below, cools against the seawater, and hardens into new oceanic crust. This process, called seafloor spreading, has been occurring continuously for hundreds of millions of years. It is the mechanism by which the Atlantic Ocean has grown from nothing to its current width over roughly 180 million years. It is happening right now, as you listen to this, at rates of a few centimeters per year, building new ocean floor that will slowly migrate away from the ridge and eventually be subducted back into the earth at the far edges of the ocean basins. The mid-ocean ridge is arguably the most geologically active environment on Earth. It is the birthplace of the oceanic crust that forms the floor of every ocean. It is the site of continuous volcanism occurring at depths where the pressure of overlying water prevents the eruptions from producing anything like the dramatic explosions we associate with surface volcanoes. Instead, the lava oozes out and solidifies into characteristic rounded shapes called pillow lavas, building mounds and flows that slowly accumulate along the ridge crest. And scattered along this 65,000-kilometer chain of volcanic activity, wherever cracks and fissures in the new crust allow seawater to penetrate into the rock and interact with the hot material below, there are hydrothermal vents. The discovery of hydrothermal vent communities in 1977 is among the most significant scientific events of the 20th century. Not because it added information to existing categories of knowledge, but because it created entirely new categories that had not previously existed. It revealed that life could exist in conditions so different from anything previously studied that it required a fundamental revision of what we thought life needed. The team that made the discovery was not looking for life. They were aboard the research submersible Alvin, studying seafloor spreading at the Galapagos Rift in the eastern Pacific. They were geologists and geophysicists examining the structure of the newly forming ocean crust. They had no particular expectation of encountering biology. At a depth of approximately 2,500 meters, in complete darkness, at temperatures just above freezing, they found something that should not have been there by any understanding available in 1977. Clustered around cracks in the seafloor, where warm, mineral-rich water was seeping upward, there was life. Not microbial life. Not sparse, barely surviving organisms clinging to existence in marginal conditions. A thriving, dense, complex community of organisms, many of them large, all of them unknown to science, living in what had been assumed to be a biological desert. Giant tube worms, some exceeding 2 meters in length, extended their bright red plumes into the water column above the vents. Clams as large as dinner plates lay scattered across the seafloor. Mats of white bacteria carpeted the rocks around the vent openings. Crabs picked their way through the community. Fish hung in the water above, feeding on smaller organisms. The scientists aboard Alvin reportedly ran out of sample containers, trying to collect everything they were seeing. Nothing they had seen, nothing that existed in the scientific literature had prepared them for this. The key question, the one that made the discovery so significant, was this: where was the energy coming from? Every ecosystem on Earth previously known to science ultimately derived its energy from sunlight. Even ecosystems that existed in complete darkness, like the communities of organisms living in deep caves or in the deep ocean sediments, depended ultimately on organic material that had been produced by photosynthesis at the surface and had sunk or been carried to depth. There was no exception. Sunlight was the foundation of all life on Earth. Full stop. The vent communities violated this rule entirely. The bacteria at the base of the vent food web were not photosynthetic. They were chemosynthetic. Instead of capturing sunlight and using its energy to fix carbon dioxide into organic molecules, they were using the energy released by chemical reactions, specifically the oxidation of hydrogen sulfide produced by the hydrothermal fluids, to perform the same biochemical trick. They were manufacturing organic matter from inorganic chemicals, using chemical energy rather than light energy. These bacteria were the foundation of an ecosystem that had no connection whatsoever to the sun, not indirectly through sinking organic particles, not through any chain of dependency that eventually traced back to the surface. The vent communities were self-sufficient, powered entirely by the heat of the Earth's interior, independent of everything happening at the surface. This was not a minor refinement of existing knowledge. This was a discovery that the boundary we had drawn around life, the assumption that all ecosystems ultimately depended on sunlight, was not a boundary at all. Life had found a way around it. Life had been doing this for a very long time, in places we had never looked, using mechanisms we had not known existed. The implications cascaded outward from this single discovery in ways that are still being explored. If life could exist independent of sunlight at hydrothermal vents, what about other environments previously assumed to be lifeless? What about the rocks far below the ocean floor, where temperatures increase with depth and water percolates through cracks in the crust, carrying chemical energy? What about the sediments of the abyssal plain, where organic matter accumulates and chemical gradients exist? What about the rocks of the deep continental crust, where temperatures and pressures are extreme but chemical energy is present? In the decades following the vent discovery, the answer to all of these questions turned out to be the same: yes. Life is present in the deep rocks below the ocean floor. It is present in the sediments of the abyssal plain, in layers that were deposited millions of years ago and have been sitting in near-perfect stasis ever since. It is present in the rocks of the oceanic crust, in fractures and pores where water and chemical energy coexist. It is present at depths and temperatures that were previously considered incompatible with biology. The biosphere, the total living portion of the Earth, is not confined to the sunlit surface. It extends downward into the crust, into the rocks, into the deep sediments. Estimates of the total mass of life in this deep biosphere vary considerably because the environments are difficult to sample. But some calculations suggest that the microbial biomass in the deep subsurface, including beneath the ocean floor, rivals or exceeds the total biomass of all surface life combined. Think about what that means. There may be as much life below the ocean floor as above it. We had no idea this was the case until the late 20th century, and we are still only beginning to understand it. The hydrothermal vent discovery was followed by the cold seep discovery in 1984. Cold seeps are areas of the seafloor where hydrogen sulfide, methane, and other hydrocarbons seep upward from the sediment, driven by geological processes rather than volcanic heat. Unlike hydrothermal vents, cold seeps do not involve elevated temperatures. The water is cold, as the name suggests. But the chemical energy in the methane and hydrogen sulfide supports chemosynthetic bacteria, and those bacteria support communities of animals comparable in their diversity and density to vent communities. Cold seeps exist on continental slopes and margins around the world, wherever the right geological conditions create upward flow of hydrocarbon-rich fluids. They are probably far more numerous than hydrothermal vents because the geological conditions that produce them are more widespread. We have found cold seep communities in the Gulf of Mexico, in the North Sea, in the Mediterranean, in the Pacific margins, in the Atlantic. Every ocean appears to have them. We have studied a small fraction of the cold seeps that exist. The organisms that live at cold seeps include some of the most unusual species on Earth. The tube worm Lamellobrachia luimesi, which lives at cold seeps in the Gulf of Mexico, has been aged at more than 250 years, making it among the longest-lived animals known to science. These worms grow extremely slowly and require the stable chemical conditions of a cold seep to survive. A cold seep community is not a temporary feature. It can persist for thousands of years, as long as the seepage continues, allowing organisms to develop on timescales that have no parallel in surface ecosystems. Methane hydrates solid crystalline structures in which methane molecules are trapped within a lattice of water ice, form in the cold, high-pressure conditions of the deep ocean floor and the underlying sediment. These structures exist across enormous areas of the ocean floor and represent one of the largest reservoirs of carbon on Earth. They are also extraordinarily sensitive to temperature and pressure changes. Warming ocean temperatures or pressure reductions associated with seafloor disturbances can cause methane hydrates to destabilize, releasing methane into the water column and potentially into the atmosphere. The scale of this methane reservoir and its potential response to climate change represents one of the most significant unknowns in climate science. We know methane hydrates exist in vast quantities. We do not know precisely how much methane they contain. We do not know precisely how they will respond to continued ocean warming. We do not know the thresholds at which large-scale destabilization might occur, or what the consequences for atmospheric methane concentrations would be. We don't know because the deep ocean floor, where the methane hydrates are, is the place we haven't looked. Beyond biology and geochemistry, the unexplored ocean contains structural features of extraordinary scale that we have only recently begun to appreciate. The seamounts, those underwater mountains rising from the ocean floor, deserve particular attention because their numbers are staggering and their ecological importance is now understood to be enormous. A seamount is typically defined as an underwater mountain rising at least 1,000 meters above the surrounding seafloor. By this definition, there are estimated to be more than 100,000 seamounts in the world's oceans. Some estimates place the total as high as 33,000 for mountains exceeding 1,000 meters. But when smaller features are included, the total count of underwater mountains likely reaches into the millions. We have studied fewer than 1% of the seamounts that are known to exist in anything approaching detail. Why do seamounts matter? Because they are, ecologically speaking, oases. In the deep ocean, where food is scarce and the sediment-covered floor offers limited structure for organisms to attach to or hide in, a seamount creates a completely different environment. Its rocky slopes, provide hard substrate. Its geometry deflects deep ocean currents upward, a process called upwelling, which brings nutrient-rich water from depth to the surface around the seamount. Its peaks, in some cases, reach into shallower and better lit water than the surrounding deep ocean floor. The result is that seamounts concentrate biodiversity. They attract fish, marine mammals, and seabirds. Their rocky slopes support corals, sponges, and other filter feeders that require hard substrate. Their elevated positions in the water column create local environments that differ substantially from the surrounding deep ocean. Many seamount species are found nowhere else. They have evolved in isolation on their particular seamount, which may be separated from the nearest similar habitat by hundreds or thousands of kilometers of open ocean over timescales of millions of years. This isolation produces endemic species, organisms that exist on a single seamount, and that would be permanently lost if that seamount were disturbed. We have no idea how many endemic species exist on the seamounts we have not studied. We have found endemic species on virtually every seamount that has been investigated in detail. The pattern suggests that the 99% of seamounts we haven't studied contain enormous numbers of species we have never encountered. The technology we use to discover seamounts is itself a relatively recent development. For most of human history, the topography of the ocean floor was essentially unknown. Depth measurements required lowering a weighted line over the side of a ship, waiting until it hit bottom, and measuring the length of line paid out. This process, called lead line sounding, could take hours for a single measurement and was impractical for systematic mapping. Ocean charts from the 19th century and earlier show the ocean floor as essentially featureless, not because it was featureless, but because no one had measured it. The invention of sonar, which uses sound waves to measure distance to the ocean floor, transformed ocean floor mapping. Echo sounders, which send a pulse of sound downward and measure the time for it to return from the bottom, allowed ships to measure depth continuously while underway. The first systematic sonar surveys of large ocean areas were conducted in the mid-20th century, and they immediately revealed that the ocean floor was not featureless at all. It was a landscape of extraordinary complexity, containing mountain ranges, trenches, vast plains, and features that had no surface equivalent. Modern multi-beam sonar systems, which send fan-shaped arrays of sound beams and can map swaths of ocean floor several kilometers wide with each pass, have greatly accelerated the mapping process. Satellites can detect subtle variations in sea surface height caused by the gravitational effects of underwater topography, allowing global-scale mapping of the general shape of the ocean floor, even in areas that have never been directly surveyed by ships. But satellite-derived bathymetric maps have a resolution of roughly 5 to 6 kilometers. They can detect a seamount the size of a large island. They cannot detect a seamount the size of a small mountain. They certainly cannot reveal what lives on any seamount or what the detailed geology of any particular feature looks like. For that, you need a ship with a multi-beam sonar and time to survey. We have surveyed, in sufficient detail, to see features smaller than a few hundred meters, roughly 20 to 25% of the total ocean floor. The remaining 75 to 80% exists on our maps as broad, low-resolution shapes smeared into smooth contours by the limitations of satellite detection. The practical consequence is that vast areas of the ocean floor contain mountains, valleys, ridges, and other features that do not appear on any map. Features that have never been seen by any instrument or any human eye. Features that may have their own ecosystems, their own biological communities, their own geological histories. We are mapping a planet and finding that our maps are wrong in the most fundamental way. They show much less than exists. The deep-sea trenches, the deepest portions of the ocean, are better known to the public than most other deep-sea environments because of their association with extreme depth. The Mariana Trench, with its Challenger Deep reaching approximately 11,000 meters, has become a cultural touchstone for the concept of ocean exploration. People know that Challenger Deep is the deepest point on Earth. Fewer people know how little we understand about it. The Hadal Zone, defined as the region below 6,000 meters depth, encompasses the deep ocean trenches found primarily in the Pacific Ocean, but also in the Atlantic and Indian Oceans. These trenches form at subduction zones, where one tectonic plate dives beneath another, creating V-shaped depressions in the ocean floor that can extend for hundreds of kilometers in length and tens of kilometers in width at their deepest points. For most of the 20th century, the Hadal Zone was assumed to be essentially barren. The crushing pressure, the near-freezing temperatures, the isolation from the productive surface waters, the scarcity of food, all seemed to argue against the possibility of a rich biological community. Early sampling efforts using dredges and simple traps retrieved some organisms, but the samples were sparse and the methodology limited. Detailed exploration of the Hadal Zone did not occur until the 21st century brought new technology and new funding. The Hadal Ecosystem Studies Project, the HADAS project, which operated in the early 2010s, conducted the most systematic investigation of Hadal environments to that point. What they found was not a barren desert. The Hadal trenches, it turned out, are excellent collectors of organic material. Their funnel-like shape, with walls rising steeply from the flat trench floor, channels organic matter that sinks from the surface into the trench. Material that might drift for hundreds of kilometers at intermediate depths before finally sinking to the abyssal plain around a trench instead gets concentrated into the trench itself. The trenches effectively vacuum up organic matter from a much larger area of the ocean above them. The result is that Hadal environments often have more food available than the surrounding abyssal plain at similar depths, not less. And where there is food, there is life. Amphipods, small crustaceans in the order Amphipoda, are the most conspicuous organisms at Hadal depths. They exist at extraordinary densities in the Hadal Zone, reaching concentrations that rival those seen in much shallower and more food-rich environments. They are efficient scavengers, able to locate and exploit organic falls, the remains of larger organisms that sink to the deep sea, within hours of the material arriving on the seafloor. Fish have been observed at depths previously considered too extreme for vertebrate life. The snailfish, a family of fish adapted to deep-sea life, have been recorded at Hadal depths exceeding 8,000 meters. These fish are translucent, gelatinous-looking creatures, quite unlike the robust forms of shallower water. Their cells and proteins are adapted to function under pressures that would destroy the biochemistry of any organism from shallower water. The microorganisms at Hadal depths are, if anything, more surprising than the larger animals. Bacteria and archaea inhabit the sediments of the deepest trenches in enormous numbers. They are not dormant. They are metabolically active, processing organic matter, cycling carbon and nitrogen, performing the same biogeochemical functions that microorganisms perform in every other ocean environment. The pressure that would crush a submarine does not particularly bother a single-celled organism evolved over millions of years to live at that pressure. In 2019, the 5 Deeps expedition reached the deepest points of all 5 major ocean basins for the first time in history. This was not a scientific expedition in the traditional sense. It was funded by a private investor, Victor Vescovo, and the scientific component was secondary to the goal of achieving the dives. But the expedition produced multi-beam sonar maps of all 5 Hadal zones at resolutions never previously achieved, and it placed human observers at depths never before visited. What the sonar maps revealed was unexpected. The Hadal trenches are not simple, featureless chasms. They have complex internal topography, with ridges, scarps, and other structural features that reflect the geological processes creating them. Biological communities are not distributed uniformly across the trench floor. They track the topographic features, clustering around structures that concentrate food or provide physical substrate. The implications are significant. If Hadal biological communities are spatially structured in ways that correspond to topographic features, then understanding those communities requires understanding the topography in detail. And the topography of most Hadal trenches has not been mapped in sufficient detail to reveal this structure. We have visited the deepest points of all 5 ocean basins. We have not mapped the detailed topography of most Hadal trenches. We certainly have not sampled the biology of most Hadal environments in any systematic way. The Hadal zone covers an area comparable to Australia. We have looked at a small fraction of it. The biological inventory of the deep ocean, to the extent one exists, is built on a foundation of deep inadequacy. The Census of Marine Life, completed in 2010, represented the most comprehensive effort ever made to catalog marine biodiversity. Over a decade, thousands of scientists from more than 80 countries contributed data and expertise. The census identified approximately 250,000 known marine species. It estimated that between 500,000 and 10 million additional species remain undiscovered. The range of that estimate deserves careful attention. Not 500,000 to 600,000, not 1 million to 2 million, 500,000 to 10 million. The uncertainty spans a factor of 20. When your uncertainty about the number of undiscovered species spans a factor of 20, you are at the beginning of understanding the system, not near the end. Every deep-sea expedition that has conducted systematic biological sampling has returned with species new to science. Not occasionally, consistently. The pattern has held for every decade since the first systematic deep-sea expeditions in the late 19th century, and it shows no signs of weakening. The rate at which new deep-sea species are being described has actually increased in recent decades as technology has improved and more expeditions have reached previously unsampled environments. This sustained rate of discovery is itself informative. In well-studied environments on land, the rate of new species discovery has slowed dramatically as biologists have accumulated knowledge over centuries. Vertebrate species, large mammals, birds, reptiles, are now rarely discovered in the terrestrial environment because the terrestrial environment has been studied extensively. The sustained high rate of new species discovery in the deep ocean tells us that we are nowhere near saturation. We are in the early phases of exploration, not the late phases. The biological communities of the deep ocean are not simply impoverished versions of shallower communities. They are distinct, with their own evolutionary histories, their own ecological structures, their own adaptations to the specific conditions of their environment. Deep-sea fish have evolved extraordinary modifications for life in perpetual darkness. Bioluminescent lures, enormous eyes, transparent bodies, expandable jaws that allow them to consume prey larger than themselves. Deep-sea invertebrates have evolved pressure-resistant biochemistry, adapted cell membranes, and modified protein structures that allow them to function at pressures incompatible with surface-adapted life. These adaptations represent millions of years of evolutionary history, occurring in isolation from the surface world, producing solutions to the challenges of deep-sea life that are often unlike anything seen in shallower environments. They are not just interesting for what they reveal about the diversity of life. They are interesting for what they reveal about the biochemical possibilities of life, about the strategies evolution deploys under different selective pressures, about the range of solutions available to organisms confronting extreme environmental conditions. Some of these solutions have proven to have applications far beyond marine biology. The heat-stable enzymes produced by thermophilic bacteria at hydrothermal vents became the basis for the polymerase chain reaction, PCR, one of the most important tools in molecular biology. PCR is used in genetic testing, forensic DNA analysis, disease diagnosis, evolutionary biology, and virtually every other field that requires the amplification of DNA sequences. The enzyme that makes PCR work, Taq polymerase, was originally isolated from a bacterium living at a hydrothermal vent. This is the pattern that the history of deep-sea exploration has established. Look, find something unexpected. Discover that the unexpected thing has implications far beyond its immediate context. The history of what we have found in the fraction of the ocean we have studied justifies enormous confidence that the fraction we have not studied contains equivalently unexpected things. We just haven't looked. The chemical oceanography of the deep ocean is another domain where ignorance is remarkable given the practical importance of what we are missing. The ocean absorbs carbon dioxide from the atmosphere through a combination of physical and biological processes. When atmospheric carbon dioxide concentrations rise, more dissolves into surface seawater. Surface organisms, particularly phytoplankton, incorporate carbon dioxide into organic matter through photosynthesis. When those organisms die, some fraction of the organic carbon they contain sinks to depth, carrying it away from the surface and slowing its return to the atmosphere. This process, called the biological pump, transfers carbon from the surface to the deep ocean, and is one of the primary mechanisms by which the ocean moderates atmospheric carbon dioxide concentrations. The biological pump has been operating continuously for billions of years. Without it, atmospheric carbon dioxide concentrations will be substantially higher than they are, and the Earth's climate will be substantially warmer. The pump is not a minor factor in the global carbon cycle. It is one of the dominant processes that has kept the Earth's climate within habitable bounds throughout most of its history. The efficiency of the biological pump depends on what happens in the deep ocean. When organic carbon sinks from the surface, it does not all reach the seafloor. Most of it is consumed by bacteria and other organisms at intermediate depths, releasing the carbon back into the water column as carbon dioxide or dissolved organic matter. Only a small fraction makes it all the way to the seafloor to be buried in the sediment, where it can remain isolated from the atmosphere for millions of years. The fraction of sinking organic carbon that reaches the seafloor, called the transfer efficiency of the biological pump, varies across ocean regions and across time. It depends on the composition of the sinking material, on the temperature and oxygen content of the intermediate water through which the material sinks, on the presence and abundance of organisms that consume it along the way. Understanding these factors and how they will change as the ocean warms is critical for predicting how the ocean's carbon absorption capacity will change in the future. Most of the processes that determine pump efficiency occur in the deep ocean, below 200 meters, in the perpetually dark water column that occupies the vast majority of the ocean's volume. This is the part of the ocean we have sampled least, the part where our understanding of what is happening biologically and chemically is most limited. We are trying to predict how a climate-critical process will respond to warming, and most of the relevant mechanisms are occurring in the part of the ocean we understand least. This is not a minor gap. It is a gap that sits at the center of some of the most consequential predictions we need to make about the future of the Earth's climate system. The oxygen minimum zones of the deep ocean provide another example of a climate-critical phenomenon that is inadequately studied. In certain regions of the ocean, particularly in the Eastern Pacific and the Arabian Sea, biological consumption of oxygen at intermediate depths exceeds the supply of oxygen from the surface, creating layers of extremely low oxygen water. These oxygen minimum zones are expanding as the ocean warms. Because warmer water holds less dissolved oxygen, and because warming may be increasing stratification that reduces the mixing of well-oxygenated surface water with deeper layers. The expansion of oxygen minimum zones has profound consequences for marine life. Many fish and invertebrates cannot survive in very low oxygen water. They are effectively excluded from expanding oxygen minimum zones, compressing their available habitat. This compression has consequences for fisheries, for the biogeochemical cycling of nitrogen and other nutrients, and for the overall structure of deep ocean ecosystems. Oxygen minimum zones also influence the carbon cycle. In low oxygen conditions, the decomposition of organic matter proceeds through different pathways than in well-oxygenated conditions, and some of these pathways are less effective at returning carbon to the atmosphere. Whether the expansion of oxygen minimum zones will increase or decrease the ocean's long-term carbon storage depends on the balance between these effects, and that balance is not well understood. These are not esoteric questions. They have direct bearing on what the ocean will look like in 50 years, 100 years, 200 years. They will determine how effectively the ocean continues to buffer climate change, how productive fisheries remain, how the chemical environment of the deep sea changes as the surface world warms. And we don't have good answers to them, in large part because the deep ocean where these processes are concentrated remains so poorly studied. Perhaps the most striking example of how the unexplored ocean matters is the polymetallic nodule situation in the Clarion-Clipperton Zone of the Central Pacific. The Clarion-Clipperton Zone is a region of the deep Pacific Ocean, roughly 4,500 to 5,500 meters deep, extending across an area comparable in size to the contiguous United States. The ocean floor of this region is covered with polymetallic nodules, roughly potato-sized lumps of minerals that have been forming on the seafloor for millions of years through the extremely slow precipitation of metal ions from seawater. These nodules are rich in manganese, nickel, cobalt, copper, and other metals. Their growth rates are among the slowest of any geological process, perhaps a few millimeters per million years. The Clarion-Clipperton Zone contains, by various estimates, more nickel, cobalt, and manganese than all known land-based reserves of these metals combined. These metals are critical components of the batteries that power electric vehicles and store energy from renewable sources. As the world transitions away from fossil fuels, demand for these metals is expected to grow dramatically. The Clarion-Clipperton Zone has accordingly attracted significant commercial interest. There are currently exploration contracts in the Clarion-Clipperton Zone held by multiple companies and national entities, issued under the oversight of the International Seabed Authority. These contracts allow the exploration of specific areas with a view to future commercial mining. The technology to mine at these depths is being developed. There is significant investment in polymetallic nodule mining as an industry that could become operational within the next decade or two. The biology of the Clarion-Clipperton Zone is not well understood. We know it is biologically rich. The nodules themselves, hard objects in an otherwise soft sediment environment, serve as substrate for organisms that require hard surfaces, corals, sponges, and other sessile invertebrates. The sediment between the nodules supports communities of organisms that live in and on the sediment, including nematodes, polychaete worms, and various crustaceans. Survey work in the zone has identified numerous species previously unknown to science. We do not know how many species exist in the zone. We do not know the degree to which different parts of the zone are connected biologically, whether the organisms in one area are the same as those in another, or whether they represent isolated populations adapted to local conditions. We do not know how quickly, if at all, the biological communities would recover from the disturbance caused by mining operations. We do not know which species are endemic to the zone and which also occur elsewhere, a distinction that determines whether mining would cause local population reductions or permanent global extinctions. We don't know these things because systematic biological surveys of the Clarion-Clipperton Zone have only begun relatively recently, and they cover a small fraction of the total area. The relationship between the unexplored ocean and human health is one that rarely enters public discussion, but it is direct and significant. The pharmaceutical potential of deep-sea organisms has been recognized since the first chemosynthetic organisms were studied in detail. Organisms living in extreme environments produce biochemical compounds that have no equivalent in surface biology. They have evolved these compounds because they needed them to function under high pressure, to stabilize proteins at near freezing temperatures, to manage the chemical stress of environments rich in toxic compounds like hydrogen sulfide. These biochemical solutions, developed over millions of years of evolution in conditions we cannot easily replicate in a laboratory, represent a library of chemical structures that human pharmaceutical research has barely begun to explore. The antibiotic crisis facing modern medicine illustrates why this matters. Bacterial resistance to existing antibiotics is growing faster than new antibiotics are being developed. The pipeline of new antibiotic compounds is inadequate to replace those that are losing effectiveness. This is not a distant problem. Drug-resistant infections kill hundreds of thousands of people annually, and projections suggest that number will grow dramatically unless new antibiotics are found. Marine organisms, particularly deep-sea microorganisms, produce compounds with antibiotic properties unlike those of any terrestrial antibiotic. These compounds have evolved to function in chemical environments very different from those in which terrestrial antibiotics operate. They target bacterial mechanisms that terrestrial antibiotics do not target. They have structural properties that make them difficult for bacteria to develop resistance to through the standard mechanisms. Several antibiotic compounds derived from marine organisms are already in clinical use or in advanced clinical trials. The deep ocean has been sampled for pharmaceutical compounds in only a small fraction of its total extent. The organisms that have been sampled are a small fraction of the organisms that exist. The compounds that have been identified and tested are a small fraction of the compounds that these organisms produce. The ocean, as a source of pharmaceutical compounds, is one of the most significant undeveloped resources in medicine, not because we have searched it thoroughly and found little, because we have barely searched it at all and already found things of genuine medical importance. Anti-cancer compounds derived from marine organisms have shown particular promise. The complexity of marine natural products, the structural diversity of compounds produced by organisms that have evolved in chemical environments, unlike any terrestrial environment, makes them a rich source of novel chemical scaffolds that can interact with biological targets in ways that simpler, more familiar compounds cannot. Several anti-cancer drugs currently in clinical use derive from marine organisms. Others are in various stages of development. The sponges and other sessile invertebrates that live on seamounts and rocky deep-sea substrates have proven particularly productive sources of bioactive compounds. These organisms cannot move when threatened or disturbed, so they have evolved chemical defenses, compounds that are toxic or repellent to potential predators. Many of these defensive compounds have turned out to have biological activity against cancer cells, against bacteria, against viruses, against the inflammatory processes that underlie many chronic diseases. Every seamount that has been studied in detail has produced new species. Every collection of organisms from deep-sea habitats has produced new compounds. The pattern is consistent enough to constitute a rule. Look carefully at the deep ocean and you will find things that have medical potential, because the deep ocean contains organisms that have been solving biochemical problems we haven't encountered for millions of years longer than humans have existed. We have not looked carefully at the deep ocean. We have looked at small patches of it. We have found compounds that are already changing medicine, and we have not looked at 95% of the available territory. The enzymes produced by deep-sea organisms represent another category of pharmaceutical and industrial potential. Enzymes are proteins that catalyze specific chemical reactions, and they are central to both biological processes and industrial chemistry. Enzymes that function under conditions different from those of typical surface organisms at high pressure, at extreme temperatures either high or low, in the presence of unusual chemical compounds, are useful for applications where standard enzymes fail. The Taq polymerase from hydrothermal vent bacteria made the polymerase chain reaction possible, but it was the first enzyme isolated from a deep-sea organism. It was not the last. Deep-sea organisms produce enzymes stable at low temperatures that are useful in food processing, in laundry detergents, in pharmaceutical synthesis, and in medical diagnostics. They produce enzymes that function in unusual chemical environments that are useful in industrial processes where standard enzymes would be denatured or inhibited. The industrial enzyme market is enormous, spanning food processing, textile manufacturing, paper production, biofuels, pharmaceuticals, and many other sectors. Deep-sea enzymes represent a largely unexplored portion of this market because deep-sea organisms have barely been screened for useful enzyme activities. The screening that has been done has been productive. The screening that has not been done represents an open-ended resource of unknown but potentially enormous value. There is a broader point here that transcends any specific application. The deep ocean contains biological solutions to problems that have existed for hundreds of millions of years. The organisms that live there have been working on those problems, refining their biochemical approaches, exploring the available chemical space for timescales that make human civilization look like an afternoon. The results of all that evolutionary work are encoded in the chemistry of deep-sea organisms. Some of it we will find useful for medicine, for industry, for materials, science. Some of it will teach us things about biochemistry that change how we understand life itself. Some of it we cannot anticipate at all, because we do not yet know enough to know what questions to ask. This is the nature of exploration. You do not know what you will find before you look. That is precisely what makes looking worthwhile. The deep ocean is full of answers to questions we have not yet asked. It is full of solutions to problems we have not yet defined. It is full of examples of what is possible in biology and chemistry that we have not yet seen, because we have not yet been to most of the places where those examples exist. Going there is not merely a scientific curiosity. It is a practical necessity, given what previous exploration has already found, and the strong expectation, grounded in that experience, that further exploration will find more. We are choosing not to go, not in any comprehensive way, not at the scale the problem demands. We are sending occasional expeditions, studying scattered locations, collecting sparse samples. And with each expedition, with each sample, with each dive, we find something unexpected that justifies going more. The argument for deep ocean exploration is not abstract. It is grounded in decades of experience that consistently confirms the same message. The deep ocean contains more than we thought, and what it contains matters. We know this. We have known it since 1977, when the first hydrothermal vent community demonstrated that our assumptions about the limits of life were wrong. We have been repeatedly reminded of it every time a careful survey of any deep-sea environment has been conducted. And still, 95% of the ocean floor has never been seen. Not because we cannot go there, because we have not chosen to. That choice, made implicitly over decades of funding priorities and research directions, has consequences. It means that the most important decisions being made about the future of the deep ocean, decisions about mining, about the disposal of waste, about the management of fisheries that depend on deep-sea food webs, about the protection of ecosystems that may be unique and irreplaceable, are being made without the information that would allow those decisions to be made wisely. It means that the medical and industrial potential of deep-sea biology remains largely untapped, at a time when both medicine and industry face challenges that the deep ocean's evolutionary library might help address. It means that our understanding of how the Earth's climate system works and how it will respond to continued human modification is limited by our ignorance of processes occurring in the part of the planet we understand least. These are not small consequences. They are among the most significant consequences of any gap in human knowledge currently in existence. The ocean that covers 71% of the Earth's surface, that contains 97% of the Earth's water, that has been running the planet's chemistry and climate for 4 billion years, that harbors more biodiversity than any terrestrial environment, that has been the stage for evolutionary experiments of extraordinary creativity and strangeness, deserves to be known. We are beginning to know it. The tools are improving. The expeditions are becoming more capable. The scientific community's appreciation of the deep ocean's importance is growing. There are reasons for cautious optimism that the age of serious deep ocean exploration is approaching, but it has not yet arrived. The 95% is still there, still dark, still unexplored, still full of organisms that have never been observed, running biochemical processes that have never been characterized, in communities whose ecological structure has never been mapped. Right now, as you listen to this, somewhere on the dark ocean floor, a creature is moving through water that has never been touched by sunlight. It is doing something, engaging in some behavior, producing some chemical compound, playing some role in some ecosystem that no human being has ever witnessed. It has been doing this in one form or another for longer than our species has existed. It will continue doing it, whether we study it or not. The question is whether we will ever know what it is doing, whether we will ever understand the world it inhabits, whether the choices we make about the use of the deep ocean will be made with any real understanding of what we are affecting, or whether they will be made, as so many choices about the ocean have been made, in ignorance of the consequences. The answer depends on whether we decide that the 95% is worth knowing. Whether the largest unexplored territory on Earth deserves the attention of curious minds and the resources of serious science. Whether we will choose to look, finally, at the place we have been ignoring for so long. The ocean has been patient. It has been waiting in the dark for 4 billion years. It will wait a little longer, but not forever. Some of what it contains is being altered right now by warming waters and changing chemistry, and the first commercial interests beginning to look at the deep seafloor as a resource to be extracted rather than a world to be understood. The window for undisturbed exploration is not permanently open. The 95% is there. It will not always be there in the form it exists today. The question is whether we will look before the chance is gone. There are proposals to mine this region before these questions have been answered. This is the clearest expression of why the unexplored ocean matters in immediate, practical terms. We are approaching decisions that could permanently alter an ecosystem we do not understand, in search of resources we believe we need, operating on the basis of biological knowledge that covers perhaps a few % of the area we intend to affect. The history of terrestrial resource extraction offers many examples of ecological disruption caused by inadequate knowledge of the systems being disturbed. Mining, logging, agriculture, and urban development have all caused environmental damage that was not predicted because the affected systems were not adequately studied before exploitation began. The deep sea offers new opportunities to make the same mistake on a larger scale, in systems that may be even less resilient to disturbance, in conditions where recovery, if it occurs at all, may take timescales measured in centuries or millennia. The nodules that would be collected by mining operations took millions of years to form. The communities of organisms living on and around them evolved over millions of years in the stable conditions of the deep sea. These are not systems that will bounce back in years or decades if disturbed, yet we are considering exploiting them before we have studied them. The instrumentation available for deep-sea research has improved dramatically over the past few decades, and the improvements continue. Remotely operated vehicles, ROVs, are now far more capable than the first systems deployed in the 1980s. They can remain at depth for extended periods, carry sophisticated sensor packages, collect carefully targeted samples, and transmit high-definition video to the surface in real time. Autonomous underwater vehicles, AUVs, can range independently through the water column and along the seafloor, mapping topography and collecting environmental data over areas too large for tethered systems to cover efficiently. New imaging technologies allow deep-sea organisms and environments to be documented in ways that were not previously possible. Acoustic systems can detect and track individual organisms in the water column. Molecular techniques allow genetic material collected from water samples to be analyzed for the presence of organisms that were never directly observed, giving a picture of biological diversity that physical sampling alone cannot achieve. Environmental DNA methods, eDNA, which detect genetic material shed by organisms into the surrounding water, are beginning to be applied to deep-sea environments with remarkable results, revealing the presence of species that would never have been found by conventional sampling. These technological advances are making it possible to study the deep ocean more efficiently and more comprehensively than was previously achievable. They are beginning to close the enormous gap between what we have observed and what exists. But technology, however advanced, cannot substitute for the fundamental commitment of resources and attention that genuine deep ocean exploration requires. The ocean is vast. The tools available to study it are capable but finite in their reach. Making meaningful progress on the 95% that remains unexplored requires sustained investment in research vessels, in instrumentation, in the training of ocean scientists, and in the long-term expeditions that allow systematic rather than sporadic coverage of deep-sea environments. That investment has not been made at the scale the problem demands. The global fleet of research vessels capable of deep-sea work is small. The number of operational ROVs available for scientific research is modest relative to the scale of what needs to be studied. Funding for deep-sea research is a fraction of what is spent on space exploration, despite the deep ocean being closer, more directly relevant to human welfare, and in many ways more accessible. This is not a criticism of space exploration, which has generated scientific returns of extraordinary value and will continue to do so. It is an observation about priorities. We have chosen, as a scientific community and as a society, to invest heavily in understanding environments that are hundreds of millions of miles away, while largely neglecting the environment that covers 71% of the surface of the planet we actually live on. The consequences of this choice accumulate year by year. Decisions are made, commercial and governmental, about the use of the deep ocean based on inadequate knowledge. Species are lost, or at risk of being lost, before they are ever discovered. Climate models are built on incomplete understanding of processes occurring in the deep ocean. And the basic scientific picture of what life on Earth is, how it is distributed, how it functions, what it has achieved in the billions of years of evolution in conditions we have barely explored, remains fundamentally incomplete. Consider what we know about the surface world versus what we know about the deep ocean. We have cataloged approximately 8.7 million species of life on land, in freshwater, and in the surface ocean. We have studied the ecology of most major terrestrial ecosystems in considerable detail. We understand the biogeochemical cycles of terrestrial environments well enough to predict with some confidence how they will respond to climate change. We have mapped every significant surface feature on Earth, every mountain range, every river basin, every major ecosystem boundary, at resolutions that allow meaningful comparison across regions. For the deep ocean, we have none of this. We have not cataloged the species that live there. We have not studied the ecology of most deep-sea ecosystems in any detail. We do not understand the biogeochemical cycles of the deep ocean well enough to predict how they will respond to change. We have not mapped the seafloor at sufficient resolution to identify most of the features that exist there. We are, in terms of our knowledge of the deep ocean, at approximately the stage that European naturalists were at with the terrestrial world in the early 18th century, when the great systematic surveys of land-based biology and geology were just beginning. The age of ocean exploration has not yet arrived. We are still at the preliminary stage, the stage of finding that there is much more to find. This fact can be read as a failure, and in some ways it is. We have had the technology to study the deep ocean in basic ways for half a century or more. We have chosen not to deploy that technology at the necessary scale. The ignorance of the deep ocean is not forced on us by technological limitation alone. It reflects a choice, or rather a series of choices, about what was worth knowing. But the same fact can be read as an opportunity, and in many ways, it is that too. The most important discoveries in the history of ocean biology have been made in the last 50 years, in the fraction of the ocean that has been studied. The remaining 95% offers the prospect of discoveries of equal or greater significance, discoveries that will reshape our understanding of life's diversity, of Earth's geological and chemical processes, of the limits of biological possibility. Every time we have sent instruments into the deep ocean, we have found something that changed our understanding of what was possible. Not occasionally, not sometimes, but every time. The pattern has held without exception across decades of deep-sea exploration. There is no reason to believe it will not continue. The siphonophore discovered in 2021 inches the Indian Ocean, a colonial organism potentially exceeding 45 meters in length, is not an anomaly. It is a data point in a consistent pattern. The deep ocean contains organisms we have never seen, exhibiting behaviors we have never observed, organized into communities we do not understand, performing functions in the Earth's biogeochemical cycles that we have not yet characterized. They are down there right now, living in darkness, under pressures we can barely generate in the laboratory, eating in ways we did not know were possible until a few decades ago, having evolved for millions of years in isolation from the surface world. And 95% of their world has never been visited by anyone. This is the truth about the unexplored ocean. Not just that it is large, though it is. Not just that it is deep, though it is. Not just that the technology to study it is expensive and difficult to operate, though it is. The truth is that we assumed it was empty, and we acted on that assumption for centuries. We looked elsewhere. We pointed our instruments upward, toward space, and outward, toward the surface world, and told ourselves that the dark water below was not worth the trouble. We were wrong. The deep ocean is not empty. It has never been empty. For the entire 4 billion years that liquid water has existed on this planet, something has been living in that darkness. Something has been evolving, diversifying, building ecosystems from the chemical energy of the Earth's interior, filling every available niche with life adapted to conditions we are only beginning to understand. We missed it. We spent the better part of human history ignoring the majority of the living space on the planet we inhabit. But we are starting to look, and what we are finding in the small fraction of the ocean we have managed to examine in any detail is already changing the story of what is possible. It is already changing the story of where life can exist, what forms it can take, what chemical strategies it can deploy, how large an organism can grow, and how long it can live. What the remaining 95% contains, we do not know. We have hints. We have patterns. We have the consistent experience of finding something unexpected every time we go down. We have the knowledge that every environment we have studied closely has revealed communities more complex and diverse than any prior estimate suggested. We have every reason to believe that the unexplored ocean is not a void, it is a presence. An ancient, complex, biologically rich presence that has been developing for billions of years in conditions we have barely begun to characterize. It is down there right now. It has always been down there. We just haven't looked. The ocean is not empty. The ocean has never been empty. And the 95% of it that no human eye has ever seen is waiting in the dark for the first light of our curiosity to finally reach it. If this journey moved something in you, consider subscribing to Sleep on Deep. Every week, we descend a little further. The ocean is waiting. The story of how we came to ignore the deep ocean is itself a story worth understanding because it reveals something important about how scientific assumptions shape what gets studied and what gets overlooked. For most of human history, the ocean was a surface to be crossed, not a volume to be explored. Sailors and navigators cared about winds, currents, and the position of coastlines. The water below the keel was relevant only insofar as it was deep enough not to ground the vessel. What lay at the bottom was irrelevant to the business of getting from one port to another. The first serious scientific expeditions to study the ocean as a scientific object rather than a navigational challenge did not occur until the 19th century. The most famous of these, the voyage of HMS Challenger from 1872 to 1876, is often described as the founding expedition of modern oceanography. Over 3.5 years, the Challenger circumnavigated the globe, taking soundings, collecting water samples, dredging the seafloor, and measuring temperatures at various depths. The expedition returned with more information about the deep ocean than had been accumulated in all of prior human history. It also returned with organisms. The dredges that scraped the seafloor in the deep ocean were expected to come up empty, or nearly so. The leading scientific opinion of the day, promoted by the eminent biologist Edward Forbes, held that life in the ocean diminished with depth and became effectively absent below 550 meters, a depth Forbes called the Azoic zone, meaning without life. Forbes was wrong, as the Challenger immediately demonstrated. The dredges that scraped the deep sea floor came up with organisms at every depth sampled, including depths far exceeding Forbes's proposed Azoic zone. Life was present in the deep ocean. The Azoic hypothesis was dead within a few years of its most prominent defense, but its ghost persisted for a century. The discovery that life existed at all depths in the ocean did not immediately translate into sustained investigation of what that life was, how it was organized, or what role it played in the ocean's ecology and chemistry. The deep ocean was acknowledged to be inhabited, but the inhabitants were assumed to be sparse, simple, and relatively unimportant. The productive biology of the ocean was understood to occur at the sunlit surface. The deep was a repository, a place where things sank and were processed slowly. It was not a center of biological activity. This assumption, softer than Forbes's Azoic hypothesis but similarly wrong, persisted through most of the 20th century. It shaped funding priorities. It shaped the questions researchers asked. It shaped the design of instruments and expeditions. If the deep ocean was a biological desert with a few strange sparse inhabitants, there was little point in investing heavily in studying it. The Challenger dredges had retrieved organisms, but dredging is a blunt instrument. It captures what happens to fall into a towed net dragged across the bottom, and it provides no information about the spatial distribution of what is captured, the behavior of organisms in their natural environment, or the structure of communities. The organisms retrieved by dredging looked strange and sparse, partly because dredging is not a good way to sample deep-sea communities, partly because many organisms are not well captured by nets, and partly because the assumption of sparsity led researchers to interpret sparse samples as confirming sparse populations rather than reflecting the limitations of the sampling method. The development of direct observation technology, submersibles capable of carrying human observers to the deep sea floor, and later remotely operated vehicles capable of transmitting live video to the surface, transformed the picture. When scientists could actually see the deep sea floor, rather than inferring its properties from what happened to come up in a net, they found something different from what the dredge-based literature suggested. They found communities, structured biological assemblages with multiple species interacting in recognizable ecological ways. They found organisms engaged in behaviors that could not have been inferred from dead and damaged specimens hauled up in dredges. They found spatial patterns, with some areas of seafloor densely populated and others sparse, in ways that corresponded to the local availability of food and the physical structure of the substrate. The deep seafloor was not a uniform sparse community of simple organisms. It was a mosaic of different communities, each adapted to local conditions, each with its own ecological dynamics. And the hydrothermal vent discovery in 1977 demonstrated that some parts of the deep seafloor supported communities as dense and complex as any in the surface ocean. The slow accumulation of these observations gradually revised the scientific picture of the deep ocean. But the revision has been incomplete because the observations have been so few. We have looked at isolated patches. We have visited specific locations, sampled specific communities, studied specific organisms. We have not achieved the systematic, comprehensive coverage that would allow us to understand the deep ocean as a whole, to map its biological communities across their full extent, to understand how they vary across ocean basins and across depth zones, to characterize the biogeochemical processes they mediate. The patches we have studied have been extraordinary. They have repeatedly contained things we did not know existed, performing processes we did not know were occurring, at scales we did not anticipate. The consistent lesson is that the deep ocean is more inhabited, more productive, more complex, and more important than was assumed. What the patches we have not yet studied contain, we can only guess. But the pattern of every study we have conducted suggests the answer is more than we expect. The bioluminescence of the deep ocean deserves particular attention because it represents one of the most spectacular phenomena on Earth, and one that almost no human being has ever witnessed directly. Bioluminescence, the production of light by living organisms, is not rare in the deep ocean. It is the rule. Estimates suggest that approximately 76% of deep-sea organisms produce light at some point in their lives. In the twilight zone, the region between 200 and 1,000 meters depth, where some light penetrates but photosynthesis is impossible, and in the midnight zone below, bioluminescence is the dominant form of illumination. In the absolute darkness of the deep ocean, the only light that exists is produced by life itself. The biochemistry of bioluminescence involves the reaction of a light-producing molecule called luciferin with oxygen, catalyzed by an enzyme called luciferase. This reaction produces light with very little heat, making it extraordinarily efficient compared to any artificial light source. The light produced by different organisms varies in color, intensity, and pattern. Some organisms produce continuous glows. Others produce rapid flashes. Some can control the timing, duration, and pattern of their flashes with remarkable precision. Deep-sea organisms have evolved bioluminescence for multiple purposes, and understanding these purposes reveals the ecological complexity of an environment most people never consider. Predators use bioluminescence to attract prey. The anglerfish, perhaps the most iconic of deep-sea creatures, dangles a bioluminescent lure above its cavernous mouth. Smaller organisms, attracted by the light in the surrounding darkness, swim toward it and into the anglerfish's jaws. This strategy works because light in the dark deep ocean is an attention-getting signal, and organisms that respond to light signals are evolutionarily rewarded when the light indicates the presence of bioluminescent food organisms. Prey organisms use bioluminescence in counterintuitive ways to escape predators. Some deep-sea shrimp produce intense bioluminescent flashes when threatened, blinding, or distracting predators long enough to escape. Some squid expel clouds of bioluminescent material, equivalent to the ink clouds of shallower squid but made of light rather than pigment, creating a glowing decoy that attracts the predator's attention while the squid escapes in darkness. The counter-illumination strategy, used by many midwater organisms, is among the most sophisticated uses of bioluminescence. Predators hunting from below have the advantage of looking upward against a relatively bright background, the sunlit water above, while their prey is silhouetted against that brightness. Many midwater fish and squid counteract this by producing bioluminescent light from their undersides, matching the intensity and color of the downwelling light from above, and effectively erasing their silhouette. A predator looking up at one of these organisms sees not a dark shape against a bright background, but a continuous field of light that provides no target. This is not a simple on-off light switch. The light produced must match the ambient downwelling light in intensity and color, and those parameters change continuously as the organism moves up and down in the water column and as cloud cover and sun angle change above. The organisms that use counter-illumination have sensory systems that measure the ambient light and adjust their bioluminescent output to match it dynamically. We know about counter-illumination. We have studied the mechanisms in detail in a handful of species. We do not know how widespread the strategy is, how variable the mechanisms are across different lineages, or how effectively different deep-sea predators have evolved to see through it. Bioluminescence is also used for communication, including reproduction. Some species use species-specific patterns of bioluminescent flashing to attract mates in the dark ocean. The patterns must be distinctive enough to be recognized by conspecifics and different enough from those of other species to avoid confusion. In environments where multiple bioluminescent species coexist in the same water column, the specificity of these signals and the precision with which organisms recognize and respond to them represent communication systems of considerable sophistication. We have observed bioluminescent communication in a few deep-sea species. We have almost certainly not observed it in most species that use it. The visual ecology of the deep ocean, the study of how organisms see and are seen in an environment where the only light is biological, is a research field that has barely begun. The eyes of deep-sea organisms have evolved under selection pressures that have no parallel in the surface world. Some deep-sea fish have eyes so sensitive that they can detect single photons. Some have evolved multiple layers of visual pigments to maximize their ability to detect the specific wavelengths of bioluminescent light produced by the organisms they hunt. Some have eyes oriented upward to detect the silhouettes of organisms above them against the faint, downwelling light, while also having eyes or photoreceptors oriented in other directions to detect bioluminescence from any direction. The diversity of visual adaptations in the deep sea reflects the diversity of visual environments at different depths and in different regions. The visual world of a fish at 500 meters depth, where a dim blue light still filters down from above and bioluminescent flashes are a constant feature of the visual environment, is completely different from the visual world of a fish at 3,000 meters, where there is no downwelling light at all and the only visual input is bioluminescent signals from other organisms. We have studied the visual systems of a handful of deep-sea species in the detail necessary to understand their function. The deep ocean contains thousands of species with eyes adapted to conditions we have not yet characterized. The diversity of visual solutions that evolution has produced in this environment remains almost entirely unknown. The sound environment of the deep ocean is another largely unexplored dimension of a world that most people imagine as silent. The deep ocean is not silent. It is, in fact, a remarkably acoustic environment. Water conducts sound far more efficiently than air. Low-frequency sounds travel enormous distances through the ocean, attenuating far less rapidly than they would in air. The deep sound channel, a layer of water at intermediate depth where the speed of sound reaches a minimum, acts as a waveguide, trapping low-frequency sounds and allowing them to travel thousands of kilometers with minimal loss. Marine mammals exploit this acoustic environment for communication and navigation. The songs of humpback whales can be detected thousands of kilometers from the singers. Blue whale vocalizations, which occur at frequencies below the range of human hearing, travel even farther. The deep sound channel has been used by military operators to track submarine activity across entire ocean basins. It was also used in early studies of long-range acoustic propagation that revealed how efficiently sound travels through the deep ocean. Deep-sea fish produce sounds. Many species of fish have swim bladders that can be vibrated to produce acoustic signals. In shallower waters, this is well documented. Numerous fish species produce sounds associated with territorial defense, reproductive behavior, and group coordination. In the deep ocean, acoustic communication among fish is assumed to occur, but is poorly documented because recording the acoustic environment of the deep sea at sufficient resolution to attribute sounds to specific species requires technology that has only recently become available. The soundscape of the deep ocean, meaning the total acoustic environment, including all biological, geological, and physical sound sources, is largely uncharacterized. We know that it exists and that it is complex. We do not know in detail what it contains, how it varies across ocean regions and seasons, or how it is structured by the presence of different biological communities. We also know, from the limited monitoring that has been done, that human activities have fundamentally altered the acoustic environment of the ocean. Shipping, which has grown dramatically over the past century, produces a constant background noise of low-frequency sound that pervades the world's ocean basins. This noise occupies the same frequency ranges used by many marine mammals for communication. There is evidence that it interferes with whale communication over the long distances that whales use for finding mates and maintaining contact between individuals. There is evidence that chronic noise exposure affects the behavior of fish and invertebrates. The magnitude of this impact is difficult to assess, precisely because we do not have good baseline data on what the acoustic environment of the ocean was like before industrialization, and we do not have comprehensive data on how it has changed since. The ocean's acoustic environment is another dimension of a system we are altering before we have adequately characterized what we are altering. The deep-sea sediments deserve attention that they rarely receive because they contain an archive of Earth's history that is irreplaceable and largely unread. As material falls through the water column and accumulates on the ocean floor, it builds up in layers that record the conditions of the ocean and atmosphere at the time of deposition. The shells of microscopic organisms called foraminifera, which live in the surface ocean and sink when they die, preserve in their chemistry a record of the temperature and chemistry of the seawater in which they lived. The composition of sediment layers records changes in productivity, in the input of material from land, in the chemistry of the deep ocean, in the patterns of ocean circulation. Deep-sea sediment cores, cylinders of sediment extracted from the seafloor using long tubes pushed into the bottom from a research vessel, have been among the most powerful tools in reconstructing Earth's climate history. By analyzing the foraminifera and other materials preserved in sediment cores, scientists have reconstructed ocean temperatures, circulation patterns, ice volumes, and atmospheric greenhouse gas concentrations over millions of years. These reconstructions provide the empirical foundation for understanding how the climate system responds to different boundary conditions, which in turn informs predictions about how it will respond to current human-caused changes. But the sediment record is distributed across the entire ocean floor, and we have sampled only a tiny fraction of it. Sediment cores have been collected from thousands of locations around the world, an impressive number in absolute terms, but a sparse sample given the scale of the ocean floor. Large regions of the deep ocean floor have never been cored. The patterns of climate variation recorded in those regions are unknown. The history of ocean circulation in those regions, the history of productivity, the history of ice volume, all of it is contained in the sediment archive, and all of it is unread. The International Ocean Discovery Program and its predecessor programs have spent decades systematically collecting sediment cores from diverse locations around the world. They have built a library of cores that represents one of the most valuable scientific archives in existence. But the library is incomplete by any assessment. The gaps in coverage leave entire chapters of Earth's climate history unknown. These are not merely scientific gaps. The climate record preserved in deep-sea sediments is essential for calibrating the models we use to project future climate. The better we understand how the climate system has behaved in the past, across a wide range of conditions, the better we can test and improve the models that project how it will behave as greenhouse gas concentrations continue to rise. The sediment archive in the deep ocean is the best record we have of past climate. The portions of that archive we have not yet read limit our ability to understand both the past and the future. The deep ocean is also the place where some of the most extreme evolutionary experiments in the history of life have been conducted. Life has been evolving in the deep ocean for as long as there has been a deep ocean, which is to say, for most of the history of complex life on Earth. The deep-sea environment is ancient, stable in some of its fundamental physical properties, and isolated in important ways from the dramatic perturbations that have repeatedly reshaped surface ecosystems. Mass extinctions that wiped out the majority of surface life had their effects on the deep ocean too, but the deep ocean's isolation from the surface and its chemical self-sufficiency meant that at least some deep-sea communities survived events that devastated the surface world. This long evolutionary history, conducted in conditions of extreme pressure, complete darkness, cold temperatures, and in some cases extreme chemistry, has produced organisms that are, in many ways, more alien than anything astronomers have proposed as potential life on other planets. The barrel-eye fish, known scientifically as Macropinna microstoma, has eyes that are oriented upward within a transparent, fluid-filled shield on top of its head. The eyes can rotate forward to look ahead when needed, but their default orientation is upward, scanning for the silhouettes of prey against the faint, downwelling light from above. The fish itself is largely transparent. It was not until 2009, when a remotely operated vehicle captured video of living barrel-eye fish in their natural environment, that scientists realized the full extent of the fish's unusual visual anatomy. Previous preserved specimens had collapsed the transparent shield, obscuring the true configuration of the eyes. The Vampiroteuthis infernalis, whose name translates as vampire squid from hell, is not actually a squid. It is the sole surviving member of a separocephalopod order that diverged from the squid and octopus lineages hundreds of millions of years ago. It lives at depths of 600 to 900 meters in the oxygen minimum zone of the deep ocean, in conditions that would be lethal to most marine organisms. It has blue blood, which carries oxygen more efficiently than red blood in low oxygen conditions. It has the largest eyes relative to body size of any animal on Earth. When threatened, it turns its cloak-like webbing inside out to surround itself with a mass of bioluminescent tissue, creating a display that may confuse or deter predators. The Vampiroteuthis feeds not on active prey, which is the strategy of its cephalopod relatives. But on marine snow, the continuous rain of organic particles that falls through the water column from the productive surface waters above, it catches this material using long filamentous structures and transfers it to its beak. A cephalopod, a group known for active predation and intelligence, reduced by millions of years of evolution in the food-poor oxygen minimum zone to a filter feeder living on scraps. The giant isopod, Bathynomus giganteus, looks like a woodlouse the size of a large cat. It is a crustacean, related to the tiny isopods found under rocks in gardens around the world, but evolved in the deep sea to sizes exceeding 40 centimeters in length. Giant isopods are scavengers of the deep sea floor, among the first organisms to arrive at the carcasses of large animals that sink to the deep sea, able to detect the chemical signals of decomposing tissue from considerable distances. They are capable of consuming enormous quantities of food very rapidly and then fasting for years. Specimens have survived in captivity for years without eating anything at all. The Dumbo octopus, named for its ear-like fins that it uses for locomotion, is among the deepest living of all octopus species. It lives at depths exceeding 4,000 meters in some species, moving slowly through the water column above the seafloor with a graceful undulating movement of its fins. It engulfs prey whole rather than using the grasping arms typical of shallower octopus relatives. It has been observed at depths of 7,000 meters, making it the deepest observed octopus and one of the deepest observed complex animals of any kind. These are organisms we know about. We have observed them. We have names for them. The deep ocean contains organisms we have not yet observed, not yet named, not yet begun to understand. The consistent experience of deep-sea biologists is that systematic surveys of any deep-sea environment produce significant numbers of species new to science, not one or two unusual specimens. Substantial fractions of the total fauna, organisms that have never been seen before, that represent previously unknown lineages, that fill ecological roles that had not been documented. The deep ocean is a museum of evolutionary experiments, many of which we have never seen. It is a record of what life has achieved over billions of years in conditions we are still learning to characterize. Every organism we discover there adds to our understanding of what is possible, of what evolution can produce under the right conditions, of how life adapts to environments that seem, from our surface-centric perspective, impossibly hostile. They are not impossible. They are inhabited, and 95% of their world remains unseen. To be human is to be curious. Looking at space is curiosity. Looking at the ocean is the same curiosity. But we built telescopes for one and spent billions of dollars sending instruments to distant planets. For the other, we have barely looked. That was a choice, and that choice has a cost. The deep ocean is not waiting for us to be ready. It has been there for 4 billion years, building ecosystems in the dark, evolving organisms of extraordinary complexity and strangeness, cycling the chemistry of the planet, storing the carbon that might otherwise warm the atmosphere past the point of habitability. It will continue to do all of this whether we study it or not, but it will do it without us knowing what it contains, without us understanding what we might lose if we disturb it. Without us being able to draw on the biochemical and ecological knowledge it holds, knowledge that has already transformed medicine and molecular biology in the fraction of the deep ocean we have managed to study. The 95% that remains unseen is not an absence. It is a presence of staggering scale and complexity, ancient beyond our ability to fully comprehend, rich beyond any estimate we have made, running on principles we are only beginning to understand. It is down there right now. It has been there, in the dark, since before the first fish swam in the ocean above it. And it is waiting, with the patience of something that has been waiting for 4 billion years, for us to finally look. The ocean is not empty. The ocean has never been empty. And the part of it we have not yet seen is the largest unexplored territory on Earth. If this journey moved something in you, consider subscribing to Sleep on Deep. Every week we descend a little further. The ocean is waiting.